Bon matin. Alors, je vais vous introduire la première conférence de ce matin.
The first conference of the day, Cécilia Légaré. She is a doctoral student in biochemistry at University of Sherbrooke, working within the GRIM research group, interdisciplinary research group on neuromuscular diseases, and at the Ecogene 21 biocluster lab.
Donc, bonjour. Tout d'abord, je voudrais vous remercier de me permettre de...
Good morning. Thank you for giving me this chance to present my research result. Today, I will be talking about epigenetics and muscular function in DM1. DM1 is a disease with several clinical complications: cataracts, cognitive disorders, cardiac arrhythmia, respiratory deficiencies, but also I would like to mention respiratory deficiencies and cardiac arrhythmia. Now going back to genetics, we have in each of our cells DNA, which will be then transcribed into ARN and then proteins, which will allow our body to do all the necessary activities. In DM1, we have DNA. Part of it is called a gene. For DM1, the defective gene is DMPK. In that gene, we find trinucleotide repetitions. Patients suffering from DM1 will have more than over 50 repetitions. It's, however, normal to have in between 0 and 50 repetitions of that gene. As mentioned earlier, we can see the DMPK gene. Here's a repetition. The lower the number of DMPG repetitions, the lower the severe cases, but the higher the number of repetitions, the more severe symptoms will appear. Some have an interruption in the repetition. These patients, rather than having CTG's repetitions, there will be interruptions with inclusions from CCGs or GGCs within the trinucleotide repetitions. There are several phenotypes in DM1, 4 or 5, depending on the authors. There's no current consensus on this. For now, I'll look into these 4 phenotypes classified depending on the number of CTG repetitions and the age of symptom when symptoms appear. Now, we'll be looking at the adult ones, the adults CTGs over 50. One of the DM1 problems in DM1 is that there's a lot of phenotype variability between patients because of the number of CTG repetitions, as mentioned. The more the number of repetitions, the more severe symptoms. Some, however, have the same number of CTGs repetitions, but will be much less affected, while others will be much more affected. There are several mechanisms that were proposed to offer this, to address this variability problem. I'd like to look into one of them, that is epigenetics. Epigenetics means on genetics. These are modifications that will be transferred from one generation to another and will affect gene expression, the level of gene expression of ARN also, more or less ARN or proteins, but will not in itself change the proteins, just like the tone of our voice. If that gentleman says, "Come and help me," while whispering, while this one says, "Come and help me," well, we really need to be there. The content stays the same, but the tone of the voice changes the meaning. and the urgency of that situation. Epigenetics adds information on DNA and will say, well, more or less needs to be done in that case. One of these modifications is DNA modification, where a methyl group will be added. Here's one here. Here, the DNA, as mentioned previously, we can add the methyl group, and we can see that a lot of RNAs are produced. And the lower number of RNAs, the lower the number of proteins is produced. So the of methyl group DNA. So my objective was to look into the methylation of DNA of the DMPK gene on muscular and respiratory profiles of patients of adult phenotype DM1. For this, we needed 98 patients affected by DM1 of the adult phenotype. Also, we had access to blood samples and also several data collected, such as muscular profile, pulmonary functions, endocrine and cardiac functions, and cognitive profile. We also measured the length of CTG repetitions and the presence of interruptions through a small pool PCR, which I will not get into today. And then we did pyro sequencing to analyze DNA methylation, and also multiple linear regressions for statistical analyses. Here, how to measure the length of repetition of CTG and interruption. We use this method. We can see that it's very stable for sperm. You can see that there's variability on the length of repetition of CTG from one cell to another. Here's a technique to measure interruptions. If there are interruptions, we can cut at the place where these interruptions are located. One repetition CTG with no processing, then looking if we can match it, and here then we can see if it doesn't match, there's an interruption. Here's my DMPK gene, CTG repetition over there. The green and white rectangles are areas where I analyzed DNA methylation. The white rectangles showed low levels of DNA methylation and very low variability, which would not really explain the induced variability of phenotypes of patients. So we will be now focusing for the rest of the presentation on areas in green, which are the L1, 2, and 6. We assessed patients from a quantitative muscle record with microfat2. We used knee extensors and shoulder abductors, and also ankle dorsiflexors. We did a make test with Newton meter, and also it was overviewed by a standardized processes from a physical therapist. Also, we measured grip strength with a Jamar dynamometer, and it was also measured by a physical therapist, and also grip strength with a pincer meter of ABL. Then a respiratory report, which is forced vital capacity, a peak respiratory rate, maximal inspiratory pressure, maximal expiratory pressure with an with a inhaler. Respiratory therapist. We can see that patients were on average 41 years old. There was an overweighting of women as part of that sample. The average was 603 repetitions rather. 8.9% showed an interruption in the CTG repetition. With regard to muscular profiles, we can see the average and standard deviations of strength. Shoulder abductors were 40.8 on average, knee extensors 80.9, ankle dorsiflexors 14.2, grip strength 8.6, pliers strength 5.2, forced vital capacity at 82.9, peak respiratory rate 73.1, maximum and inspiratory and expiratory pressure 66.4 and 58.5. Then I made a model to based on multiple linear regression to look into the variability of muscle profile. We added variables that we thought would influence shoulder strength with sex, age, CTG, length of repetition, and then we added DNA methylation to see if it would add a layer that to the explanation of variability. As we see, DNA methylation does not explain the strength of ankle dorsiflexors. However, it can explain the strength, or rather, it cannot explain for knee or shoulder, but it can explain the strength of ankle dorsiflexors. Here we notice that L6B does not fit as part of the muscle profile variability. However, for the respiratory profile, that is the most present type of DNA methylation. It would be more present for respiratory profiles than muscle strength. Here's the same variables that were used: age, sex, length of CTG repetitions, interruption, smoking, and weight. You can see DNA methylation. We explain peak inspiratory and expiratory rates and average rates of respiration. Also, in conclusion, the DNA methylation in the locus DMPK is associated with the respiratory muscle profile of patients affected by DM1, and that independently on the length of CTG repetitions. Epigenetic markers could explain part of the phenotypical variability. We only covered part of a DMPK gene, so even though if we found some sites, probably other ones could be associated to this profile. So we would like to analyze the whole DMPK gene to see if other genes could also explain, or those sites could explain variability of DM1 phenotypes. Also, we look into DNA methylation in blood. Blood is not really affected, but we would like to measure muscle methylation. This is funded also by the Grand Défi vers la Voix Foundation. You probably wonder why this can bring in clinical terms. Well, I would like to conduct a longitudinal study to validate the possibility to predict severity and progression of DM1 using DNA methylation. Looking at if there's a certain methylation in a gene site, could we predict in X length of time such symptoms would be developed, and then maybe try to compensate or try to find a preventive treatment rather than just seeing the appearance of a symptom. Eventually, maybe we could look into DNA methylation to explain variability of other therapeutic targets if a patient answers more favorably or not to a specific drug. Well, maybe the genetic profile could partly explain the reasons why. I'd like to thank my research director, Luigi Bouchard and Cynthia Gagnon, the EpiMet team, Equigene 21 BioCluster, and the GRIM team, Darren Moncton and Gail Overen's lab, who conducted all of the CTG length and of repetitions and the presence of interruptions. And thank you very much. Do you have any questions?
Bonjour, Cécilia.
Bonjour.
Bravo pour ta présentation.
Good morning, Cecilia. I have a few questions for you. Maybe it's because I know absolutely nothing about epigenetics. You talked about the transmissibility of epigenetics phenomena. Could you elaborate on this? How is this transferred from one generation to another? Epigenetics markers are transferred during development the fetus will remodel the epigenome, but part from his parents will be kept. Studies on mice were conducted, and the epigenome will transfer from the parents of the mouse, and this will really affect the fetus. DNA methylation, is that a stable phenomenon over time? Is that because you want longitudinal studies? Answer, it can adapt depending on sites. Some sites will be affected by age, other ones will not really be affected. That is really specific, depending on the specific sites. It could, however, still be possible to look into the evolution of this over time, so we could still see evolutions between both moments. Well, that's what I think, but of course I didn't make the analyses. Another question, you talk about CTG interruptions, and that's very interesting because you're the first one looking into clinical results or clinical phenotypes with regard to interruptions in our population, but it doesn't seem that out of your regression analyses it came out that if there was an influence over this. Is it because you only have 9% of the population? There's about 9% CTG interruptions, so maybe the N number would be answer. Well, the N number is already more than what's in the general population. What in the general population is about 5% of the population will see interruptions right away. With 9% of the population, we can already have a higher sample or a higher number of N than we currently have. Also maybe in link with clinical phenotypes that were assessed. We don't really see the effect. What came out of this is the age of symptom appearance also, or onset. Some who had interruptions will have a later onset of symptoms, and this will be more effective than those who have no interruptions. Thank you.